Okay, student, whether I'm audible and screen is visible, can you tell me? So whether I'm audible and screen is visible, I will run the poll, you can tell me, okay? Yeah. So tell me whether I'm audible and screen is visible. Let us start the class. Very good. Okay. Thank you. So before starting the class, we will recall what are the things we have studied in the last class. Okay. So can you recall, guys, what are the things we have studied in the last class? Okay. Okay. So I will ask the question. You can tell me. Okay. So BT toxin, right? So BT toxin is useful for the. So which are the organisms? I have told you, right? I will give the options. You can tell me whether it is Lepidopterans, Lepidopterans or Coleopterans or Dipterans or all of the above or all of the above. Can you tell me, student, whether option A, sorry, B, C, D? So which option is correct here? Just I'm recalling. Can you tell me in the options? Or else I will run the poll. You can answer me in the chat box. Sorry, in the poll also. Which one is the correct answer, student? So BT toxin is toxic for which are the given below? Okay. Can you answer me? I didn't get answer from anyone. Lepidopterans, coleopterans, diphterans, all of the above. I need answer from you all, student. It is very easy question. JNV Chitradurga, answer ಮಾಡ್ತಿಲ್ಲ ನೀವು. Answer me. JNV Medak, answer me, students. Yeah. Okay. So the correct answer is all of the above, right? So all of the above is the correct answer. Okay. So let us talk about the application in the medicine. Okay. Yeah. So in the last class, we have studied about the application in the application of biotechnology in the agriculture, right? So let us see here. So there are so many drugs and so many vaccines are produced in the biotechnology, which is very, very useful in the medicine. Let us talk about it. Okay. So recombinant DNA technology help for the mass production of safe and more effective therapeutic drugs. Okay. Therapeutic drugs we will use for the diseases, right? Diseases to cure the diseases. The product from the non-human sources. For example, if you think the product from the non-human resources, okay, non-human sources, it can create some immunological responses or some allergic reactions. Okay, so due to that, so they will produce the transgenic animal, thereby producing the medicine, or by the recombinant DNA technology, they're used to produce the medicine. So let us take an example of insulin. In this medicine, we are taking the example of insulin. Let us study about that. See here. It is going to produce some immunological responses. So that's why we have started to produce in the biotechnological processes. Okay. So at the present, so there are 30 recombinant therapeutic has been approved actually. So but 12 are in market now. So that we are already using the 12 recombinantly produced products. Okay. So let us go to the, so where in the medicine it is going to helpful. Okay. So see here in the genetically engineered insulin, so many diabetic patients now using the insulin, right? So that is produced by the genetic engineering process. Okay. So then we'll go to the gene therapy. So what is the gene therapy? I will tell you next. And molecular diagnosis. Whenever we want to go anywhere, so they will tell now in the corona situation, they will tell RT-PCR test, right? So that those all the molecular diagnosis we are going to study in the today's class. Okay. So let us talk about the 1 minute. 1 minute. Okay. So let us talk about the genetically engineered insulin. So what we are going to do in the genetic engineering process. So student, if the insulin was not prepared, means it is very difficult to treat for the diabetic patient, right? So what is the function of the insulin? What is the function of the insulin? So where is the insulin was produced, student? Can anyone tell me? Where is the insulin was produced in our body? In the pancreas, right? In the pancreas, there is a beta Langerhans cells. Beta Langerhans cells will be there. So there, there it is going to produce the insulin, right? Insulin. So what is the function of the insulin? For example, if you see in your blood, if you have a glucose, right? If you have so much of the glucose, now only you had a lunch. So now the glucose was entered into your blood. So after the digestion and absorption, I'm telling. Okay. So now the glucose was absorbed into the blood. So now what will happen? Increases the glucose concentration in blood. So now insulin was released. Okay. This insulin, what will do? They will take this glucose molecule and they will form this glucose into glycogen. Glucose into the glycogen. Where it is going to form the glucose into the glycogen, student? Where it is going to form? In the liver. Right? So that's why so many people, secondary diabetics, the patient which are having the diabetes, they are not able to produce the insulin. Right? On that time, we are going to give a insulin from the outside. Okay. So in that case, what we have did is we started to synthesize the insulin from the pigs and the cows. Okay, from the slaughtered pig and the slaughtered pig and cow. Okay, so the problem happened was it is going to produce some immunological response. Okay, immunological response means allergic reaction. Immunological response. Response. Okay, that is nothing but some patient who took this, the insulin prepared by the slaughtered pig and the cow, they showed some allergic reaction. Okay, they showed some allergic reaction. So because it was produced in the different organism, right? So they are genetically different, right? Cow and the pig. So now what happened? So this was stopped, okay? So then they have started to produce. So student, in the bacteria, can we prepare the insulin gene? Sorry, can we prepare the insulin protein? Insulin by using the bacteria, can we prepare insulin? I need answer from you. No, right? It is not possible because you know that. So the genetic, how the processing of the genes will happen. So that is very different compared to the eukaryotes and the prokaryotes, right? So gene slicing and introns were absent in the prokaryotes, right? So that is the reason. So we will not use the prokaryotes. So that is the bacteria, but and it is not going to give a complete insulin protein. Okay. So what they have did in the next is, see here, it is not at all easy to produce complete insulin in the bacteria. Okay. So what is the process? In the after the insulin production, so it is produced as a proinsulin. So that is the inactive insulin. Okay. So whenever the insulin was produced in our body, so this is going to be insulin. 1 minute. Otherwise, you will get confused here. Whenever the insulin was produced, it is going to be produced in the proinsulin form. What is the proinsulin form, student? So it is the inactive form. Okay, inactive form. So later what will happen? So this inactive insulin, see here, this inactive insulin is consisting of the 1, 2 and 3. Okay, so one is the A chain is there, B chain is there, and the C chain is there. Okay, so one is A chain, B chain, and the C chain. See here. Okay. See here. So this is the proinsulin. Okay, proinsulin. It is having the A chain. This is the B chain. 1 minute. And this is the C chain. Okay. C chain. So this A chain is made up of the 21 amino acid and B chain is made up of the 30 amino acid. Okay. So here you can see here, this is the mature insulin. Mature insulin. Okay. This mature insulin is going to have only the A chain and B chain. Okay. There is no C chain. Okay. There is no C chain in this thing. So what will happen now? See here. So during the maturation process, this C chain was removed. Okay. During the maturation process, during maturation of insulin, C chain will be removed. Okay, C chain will be removed. Okay, this is very important. Okay, C chain will be removed. So, but this thing, this maturation process or removal of the C chain will not be happen in bacteria. Okay, so that's why the one of the company have synthesized this A chain and the B chain separately. First, we will study about the structure of the insulin. Okay, see here how the insulin structure will be between these A chain. Okay, this is the A chain. So this is the B chain. This is the mature one. Okay, so between this A chain and this B chain, there is a disulfide bond is there. Okay, but this disulfide bond will not be formed in the bacteria. So that is the one reason why we will not synthesize in the bacteria. Okay, so and 1 minute. This is a A chain and this is a B chain. And this A chain is having the intradisulfide bond. Intradisulfide bond. Okay. So between the A and B chain, there is a disulfide bond is there. Whereas the A chain is having the intradisulfide bond. Okay. So this is in the mature insulin. Okay. This is in the mature insulin. So this is not happen in the bacteria where the disulfide bond formation will not take place in the bacteria, and also the separation of C peptide will not take place in the bacteria. So that's why what the biotechnologists think is they have released one of the company. So that is in the year 1983. Okay, in the year 1983, that company name was Eli Lilly. Okay, Eli Lilly, what they have did is by in the vector they have inserted the A chain. Okay, A chain, and in the another vector they have inserted the B chain. Okay, they have inserted the B chain. Okay, so what will happen now? They will insert it into the E. coli bacteria. So now A chain was synthesized. Okay, so this is the A chain. A chain was synthesized. 1 minute. A chain was synthesized. Okay, so now what they have did is, so they have inserted this B. So this is a recombinant DNA, right? Recombinant DNA. So this is the recombinant DNA. So then they have inserted into the E. coli. So they have multiplied that and they have given the B protein. Okay, so they are given the B protein, right? So now A protein is ready. B protein is peptide, not protein. Peptide. A peptide, B peptide is ready. So now see here what they have did is they have separated the A peptide and they have taken the B peptide separately. So this is A peptide. So this is a B peptide, and they have inserted the disulfide bond separately. They have inserted this disulfide bond separately. Okay. So now the protein was obtained. That is the mature insulin was obtained. Okay. So what they have did is in the laboratory condition, they have added the disulfide bond. They have added disulfide bond separately. Right? So this is what they have did. Now we are using the insulin because of this reason. So that is they have synthesized this A peptide separate, B peptide separate. So now they have purified that and have added the disulfide bond separately. So now our insulin was prepared. So do you have any confusion here, guys? Can you tell me? Whether do you have any confusions here? I need answer from you. JMV Chitradurga, JMV Medak, can you tell me? Srinivas Ram. Okay. Let us go to the next. Okay. So what is the gene therapy? So student here you have to remember that some of the people are having some genetic disorders, right? If genetic disorder, in the genetic disorder, what will happen? So gene will be mutated or they're having some faulty genes, right? So that faulty gene, for example, if you take the phenylketonuria and cystic fibrosis, what will happen? The gene is mutated there, right? The faulty gene was there, right? So what we will do in the gene therapy is, so we will remove the faulty gene and we will add the correct gene in that place. Okay, for example, if you take, so this is the gene. Okay, this is the gene. So this is the DNA. Okay, this is the DNA. Okay, so here one gene is there. So this is mutated, or we can call it as the faulty gene. Okay, this is a faulty gene. Okay, so this gene we will remove and we will add the, for example, see here. One minute. Yes. So this gene is going to code for the ADA. What is that ADA? Adenosine deaminase. Adenosine deaminase. Okay. So this gene is code for the adenosine deaminase. This person who is having the mutation is going to have a disease that is called as the SCID. What do you mean by SCID? SCID is nothing but severe combined immunodeficiency. Severe combined immunodeficiency. Okay. So he is having the severe combined immunodeficiency. That means he is not able to synthesize the B cell and even the T cell. Okay. So not able to synthesize B cell and T cell. Then he is susceptible to all the type of the disease, all infection, right? All type of the infection, right? So that is about this gene. So now what we'll do in the gene therapy, we will remove this gene. Okay. See here, in the gene therapy, what we will do, we will remove that faulty gene and we will replace it by the correct gene. See here. So we have replaced replacing, 1 minute. Replacing faulty gene by the functional gene. Okay. Replacing faulty gene by functional gene. Okay. So that is what we will call it as the gene therapy. Okay. Gene therapy is nothing but replacing faulty gene by functional gene. Here only you can see what I have did. I have removed this faulty adenosine deaminase gene and here I have inserted the functional one, right? Functional gene, right? So this is what we will do in the gene therapy. See here, it is a method to correct the gene defect. It is a method to correct the gene defect. Okay, in the child or in a embryo. See here, genes are inserted into the person cell or the tissue to treat a hereditary disease is compensate for the non-functional gene. Okay, so here there are two types of the gene correction. Okay, first thing we will do in the somatic cell. Okay, first thing we will do in the somatic cells. Okay, so now if any person is a grown up, okay, is adult one, is having any faulty gene in his genome, means we will remove that gene and we will replace by the functional gene. So that is in the somatic cell. Okay, That is the somatic cell therapy. So the another one is there. So that is the germline gene therapy. Germ, 1 minute. 1 minute. Germline gene therapy. Germline gene therapy. So what we will do in the germline gene therapy? Germline gene therapy is related to the egg and sperm. Okay, egg and sperm. For example, if the father is having that faulty gene, faulty gene means we will remove that or replace that gene by the functional gene in the sperm. Okay. If the mother is having that faulty gene means we will take the egg and we will replace it by the functional gene. Okay. So then we will do the fertilization. Then we will do the fertilization. Then the zygote will form and the baby will born without the disease. Okay, the baby will born without disease, right? Without the disease, without disease, right? So this is what they will do in the germline gene therapy. So germline gene therapy is a permanent cure. Okay, so it will be the permanent cure for any disease. Okay, permanent cure. But now, so they are not given permission for doing the germline gene therapy. Okay, the permission is given only for the somatic gene therapy. So there is no germline gene therapy permission. Okay, they are not given the permission for the germline gene therapy. So let us go to the next. I think you understood this, student. Can you tell me whether you understood or not? Can you tell me whether you understood or not? What is the germline gene therapy? Can you answer me in the chat box? Okay. So let us go to the next. So the first patient, okay, the first girl undergo the gene therapy was the Ashanti De Silva. Okay, so she's the first person. Okay, she's the first human to undergo the gene therapy. Here you have to remember, okay, gene therapy. So in the 1990, okay, in the 1990. So when she was 4 years old. Okay, when this girl is the 4 years old. So this girl is suffering from she is having the SCID. Okay, I have told you what do you mean by SCID, right? So that is the severe combined immunodeficiency. Severe combined immunodeficiency. Immunodeficiency. So that is due to the deficiency of ADA enzyme. ADA enzyme. So that is adenosine deaminase. Deaminase. Okay. Okay. So now what will happen? So they have taken the gene from so they have collected the cell first. Okay. What is the cell they have collected? So first they have took the bone marrow. Okay. First from the bone marrow, they have collected the leukocyte. Okay. So student, what is the leukocyte? This is the WBC cell, right? This is the WBC cell. From the leukocyte, they have taken from the bone marrow, they have taken the leukocyte, WBC cell. So this WBC cell is having the deficiency, right? See here, 1 minute. So they have took the WBC cell. This is the cell you think, okay? This is the genome of the cell. It is having the faulty gene. 1 minute. So it is having the faulty gene, okay? Faulty gene. First, they will what do? They will culture this leukocyte, okay? They will culture this leukocyte in the laboratory. See here now we have so many leukocyte cell, right? So then what they will do? They will take they will remove the faulty gene. Then they will replace it by, functional gene. Replace it by the functional gene. Okay. So what they will do? Now they will again, so now they have got the, now our gene is there and our gene with the, our gene is there, our gene with the functional gene, functional, sorry, our DNA is there with the functional gene. Okay, so now what will happen? So this cell, they will insert it into the cell. Insert this into the cell, which is that cell, student? WBC, right? Leukocyte. So then this WBC cells will again, they will insert into this bone marrow. Okay, so they will insert into the bone marrow. So this is the not a permanent. Okay, this is not a permanent cure. This is not a permanent cure. I told you why it is not a permanent cure, right? Because they are inserting into the somatic cells, right? Because this WBC, when it is goes inside the bone marrow, it will die, right? So it is having some particular time, it will die after that time, right? Only till that time, it will give a, it will going to produce that ADA enzyme. After that, it will not going to produce. So for that reason, there are two methods for the treatment of this kind of the disease. So that is the ADA. Sorry, for this kind of the SCID. So first one is the bone marrow transplantation. Bone marrow transplantation. Okay, student, bone marrow transplantation. And second one is the enzyme replacement theory. Enzyme replacement, sorry, treatment, not theory. Replacement treatment. Enzyme replacement treatment. What they are going to inject? So first one is the by the bone marrow transplantation. Okay, first method is the bone marrow transplantation, and second one is the enzyme replacement treatment. So particularly, they will inject the enzyme into that bone marrow, and after that, sometimes the enzyme is going to be that enzyme is going to be removed. Means it is, that is a protein, right? It is going to be removed. So then again we have to add the that enzyme. So that is we call it as a enzyme replacement theory. So enzyme replacement treatment. Okay. So that is for this SCID. Okay. So both are not permanent. Okay. So these both are not a permanent cure. Both are not 1 minute. Both are not permanent cure. Not okay. Okay. So that is the reason what they have to do. That embryonic stage. Okay. The same thing. So that is adding the gene in the embryonic stage if we do it. So that is the permanent cure. Permanent cure. Okay. So this is about the gene therapy. If you have any questions, you can ask me. Or we'll go to the next. Okay. Yes. So then we'll go to the molecular diagnosis. So see here in molecular diagnosis, so there are the, why we will do the molecular diagnosis. So sometimes we'll come to know about the disease is there by analyzing the urine and the serum, right? So if you go to the doctor with the symptoms, the doctor will tell that if you have the symptoms, means you go for the urine test or you go for the blood test, right? Yes. So that is when we are having the symptoms, right? But here we don't have any symptom, but we are diagnosing that whether this disease is there or not. Okay. So that is what we will call it as a diagnosis. For example, if you take the RT-PCR test, okay, corona. So if the person do not showing any symptoms, but then also he will be positive for the corona, right? It will show when the viral titer was very, very less. Okay. On the time only it will show that whether the virus was there or not, right? So in the diagnosis process we will do. So that is before the symptoms only, before symptoms. Okay. See here, the conventional methods, so that are the serum and the urine analysis, are not suitable for early diagnosis of the disease, right? So when the disease was caused and they are showing the symptoms, On that time, they will do the urine and the serum analysis. Okay. Before we have to do means the molecular diagnosis was very, very helpful in that process. We'll study about that today. Okay. See here, it is possible by the technique like recombinant DNA technology and the PCR. So, student, can you tell me where we are doing the ELISA test? Can you tell me where we are going to do the ELISA test? Here you can see which are the molecular diagnosis we will do. So, do you remember PCR? Obviously, you will remember the PCR, right? Do you remember RT-PCR? Yes, you will remember because for the corona we will do the RT-PCR. And Western blotting, student? Western blotting, you remember? Or have you remember what is the Southern blotting? Southern blotting. Have you remember what is the Northern blotting? Northern blotting. See here, for the Western blotting, we will do for the protein. Okay, to check the protein. So, and for the Southern blotting is for the DNA. And Northern blotting is for the RNA. Okay, so these all the things, these are the molecular diagnosis process we will utilize. Okay, so see here they are given the recombinant DNA technology has given the PCR and the ELISA. Let us talk about that. So can you tell me what is the full form of PCR? So student, I need answer from you. JV Chitradurga, JV Medak, Srinivas Rao, Petanapati, can you tell me? Can you tell me what is the full form of PCR? I need answer from you. What is the full form of PCR? Can you tell me whether I'm audible and screen is visible? No one is answering here. Can you tell me? Can you tell me whether I'm audible and screen is visible? I need answer from you. Okay. So let us, Then answer me in the chat box. Very good, my dear student. Very good. Okay. Yeah. Thank you so much. I have to know whether you are understanding or not. So tell me in the chat box. Otherwise, I will tell you some other examples also. No. Okay. Let us go to the polymerase chain reaction. For it, what we will do in the polymerase chain reaction. For example, if you are having the coronavirus in your body, but you don't know, you don't have any symptom. On that time, what they will do? They will take a sample. Okay. Any saliva they will take, they will take the sample from the nose and the mouth, right? So see here, what they will do? They have take this sample, right? This is the sample. In the sample, only one DNA is there. See here. So this is the RNA. When it compared to the coronavirus, this is RNA. Okay. This sample is there. Only this is very less quantity. Okay. So what they will do in the PCR is they will multiply the copies. Okay. So they will do the just Xerox copy. Okay. Now you are having a one test book. You'll go to the Xerox shop and you will do the so many copies of that. Right. So this is how they will do the so many copies of that. So here, for example, one copy you have means. So here they will do the 1000 copies. 1000 copies in the PCR. So next they will what do? They will do the gel electrophoresis, right? They will do the gel electrophoresis. Gel electrophoresis. In the gel electrophoresis, after that, what they will do, student? If this is the DNA, so they will do the Southern blotting. Southern blotting. If it is RNA, they will do the Northern blotting. Okay. So now they will do the Southern blotting. Okay. So after they are doing the Southern blotting, so in the Southern blotting, what they will do is this DNA is separated. Okay. So this is a viral DNA. Okay. This is the viral DNA. We have a viral DNA before only. Okay. This viral DNA we have, and we have radio labeled this. Okay. So we have radio labeled this. So this DNA is complementary to this viral DNA. So this is what we will call it as the probe. Okay, complementary DNA. Okay, complementary DNA. So now what will happen? This DNA is going to be added to this solution now. Okay, see here, this DNA we are going to add to this solution. So now this probe is going to give. See here, this is only binded to this DNA, that is the viral DNA, right? It is not binded to this all the DNA, right? So now they will tell that this is a positive result, right? So this is how they will do by using the probe, complementary DNA. Okay, yeah. So then this is about the PCR. Okay, this is about the PCR, how they will do the PCR, polymerase chain reaction. Polymerase chain reaction, right? No. So then what they will do in the ELISA, let us see. See here, ELISA, can you give the full form of what is the ELISA, student? ELISA. ELISA is nothing but enzyme linked immunosorbent assay. Assay, right? Enzyme linked immunosorbent assay. So why they will do the enzyme linked immunosorbent assay? So do you remember in the detection of the AIDS, right? In the detection of the AIDS, they will do the ELISA test, right? Yes. So see here how they will do that. Okay. So students, see here, this is the one plate. Okay. So this is one plate. For this plate, there is already attached the antibodies. See here, this is one plate. For this plate, already they have attached the antibody. Already antibody is there. Okay, this we will call it the ELISA plate. ELISA plate. Okay. So see here, this is the primary antibody. Primary antibody. Okay. This will be specific to the antigen. Okay. This will be specific to antigen. So now what will happen? See here. Now we'll add the sample. For example, they have taken some saliva from your sample, right? Now this saliva is having the antigen. Okay. Now this saliva antigen will come and bind to this antibody paratope. This paratope we will call it, right? Paratope, right? So this is the antigen. Okay. So now what we'll do? What they will do? See here, this is the plate is there. Now our antibody is there. Our primary antibody is there. Here you can see antigen was binded to this, right? Now they will wash. Okay, they will do the washing. They will do the washing. Washing means not by using the soap and all the things. Okay, just they will do the just they will add the buffer and just they will remove the buffer. That is what we will call it as the washing. Okay. So the antigen which is not binded to this antibody will be removed by washing. So now the antigen which is specific to that antibody, that is binded, right? So now what they will do, they will add secondary antibody. They will add secondary antibody. That antibody is enzyme linked. Okay, that antibody is enzyme linked. See here, this is the antibody, right? This is the antibody. So here you can see this is a heavy chain, this is a light chain, right? So here you can see this is a constant region, right? This is a constant region. This is a variable region. This is a variable region where antigen will bind, right? For the variable region. Variable region, antigen will come and bind. So here it is the constant region. For the constant region, they have added the enzyme. Okay. So what is the function of enzyme, student? Enzyme will convert substrate into the product. Substrate into the product. For example, you can see here, this substrate is colorless. This substrate is colorless. Okay. Now, this convert into the colored product. Colored product. Colored product in presence of the enzyme. So some of them will confuse that one, what is the substrate, right? In the chemistry, you have learned about the reactant. Reactant will give rise to the product, right? So that is what? Nothing but the reactant and the product. So that is the substrate and product. Okay. So see here. Now, this secondary antibody was added, right? This we will give the secondary antibody blue color. Okay. See here. Where it will come and bind? The secondary antibody will come and bind to the antigen. See here. The secondary antibody is having the enzyme linked to this, right? Enzyme linked to this, right? So now what will happen? So our secondary antibody is there, our primary antibody is there, along with that enzyme is there. Enzyme is there, right? So that's why we'll call it as enzyme linked immunosorbent assay. Enzyme was linked to this, right? So that is the reason. So now what will happen? For this, I am going to add the substrate. See here, this is the antibody. This is the antigen we have added. So here we have added the secondary antibody, right? This secondary antibody is having the enzyme here. This enzyme is there, right? So now I will add the colorless product. Sorry, colorless substrate. See here. Now I am adding this product. Sorry, substrate. This color is not visible. 1 minute. Now I will add the substrate. Add substrate. See, a student here, you have to remember that the secondary antibody is also the specific to the antigen. After that also we will wash. Okay. So which are the secondary antibody attached to the this antigen that will only bind to the this antigen. Other antibodies which are not binded, they will remove in the washing. Okay. So now, if it is antibody, secondary antibody was attached, means it must have the specificity to that antigen, right? Then, if it is attached, means it must convert the colorless product into colorless substrate into the product, right? So that is convert the colorless substrate into the colored product, right? Colored product, right? So what it will do? So this will convert the, we will add the substrate, it will convert the colorless substrate into the colored product. So then we will tell that this is a positive result. Okay, color came means positive result. If there is no color means we will tell that negative result. Negative result. Okay, student. Now you understood what is the ELISA? Can you tell me Jainvi, Meghak and Srinivas sir and Jainvi Chitradurga, do you understood this? Okay. So then we will talk about the transgenic animals. Okay. So, okay. Very good. Very good. Okay. So student, now only we have talked about what is the how we are going to add the gene to the animals, right? For example, we have talked about the gene therapy, right? So see here, if you add the gene only in the somatic cell, okay? If we add the gene in the somatic cell, means is it going to transfer to the progeny? Is that going to transfer to the progeny, student? No transfer to the progeny, right? No transfer to the progeny. Progeny is nothing but babies. Okay, it is not going to transfer to its babies. Okay, if we do the germline, 1 minute. In the embryonic cell. Okay, so if we add that in the embryonic cell, if we add that in the embryonic cell, means it is going to show the, it is going to transfer, okay, it is going to transfer to the progeny. Okay, so this is what they will do in the transgenic animal. Okay, so what they will do, so they will do the, for example, male and female is there, right? When we are doing the, we will take the male nucleus. Okay, male nucleus we will take, and in the male nucleus we will add the gene. We will add the gene of interest. Okay, by the which method? Micro injection method. by the micro-injection method. Okay. So then this is like male pronucleus, we will call it as, okay, male pronucleus after the fertilization. Okay. So male, so the sperm and the egg is fertilized, right? So that is we call it as a male pronucleus. For this, we will add the gene and this is zygote will form it. Okay, zygote will form. So this will we are going to add into the surrogate mother. Surrogate mother to the another animal. Okay, to the another animal. One thing may sperm and egg is taken from the one animal to the same species only to the another animal for the mother for the another female. We are going to transplant this zygote. Okay, this we will do in the test tube baby. In the test tube baby, they will do no same thing. Okay, so now our gene is there. This was inserted into this animal. Inserted into this animal. And this progeny is also going to show this gene. Okay. See here. So we were, if these are the animals whose genome has been altered by introduction of an extra foreign gene by the manipulation. Okay. See here, this rat is not having the color gene, right? So now it is going to produce the radium-like light. So that is a bioluminescent light, right? So the gene was inserted into this rabbit, then it is going to produce the bioluminescent. Okay. So this is how they will introduce the foreign gene by the microinjection method. See here. So the commonly will produce the transgenic rat, transgenic rabbit, pigs, sheep, and cow. Okay. But here 95% will produce the transgenic animal. So that is the mice. Let us talk about, so which are the useful benefits of the transgenic animal. First one is regulation of the genes in the normal physiology and development. Okay, to study the normal physiology and the development. Of the gene in the development of the biological product. Third, fourth one is the vaccine testing, and fifth one is the chemical safety testing. Okay, let us study about one by one. For example, student, in the study of the normal physiology. For example, if you take the animal is there, okay, this animal, I will remove the gene for the, for example, this is the embryo of the animal. Okay, embryo. This is the embryo of the animal. Okay, so I will remove one gene. Okay, remove the one gene. Okay, after removal of that gene, the tail development was not happen. The tail development not happen. Not happened. Happened. Okay. In some, what will happen? I will take the embryo and I will remove the gene. Remove the gene. Here, brain development was not happen. Brain development retarded. Brain development not happen. Okay. So this is how they will study about the gene. What is the function of the gene? Okay. So this is by studying the gene. Okay. What they have did? They have taken the embryo. They have removed the gene. So then there is the animal was formed, but there is a no tail. Then they have removed the one gene. Thereby the brain development was impaired. So thereby studying the function of the genes. Okay. So they have used the transgenic animal. And see here, in the development of the disease. So student, you know, the cancer was produced by the two types of the gene. One is called as the proto-oncogene and another one is the tumor suppressor gene. Tumor suppressor gene, right? So whenever the mutation was happened in this gene, it is going to cause the cancer, right? So this the transgenic animal is also useful in studying this type of the diseases. So that is the cancer. So now they have studied by using this transgenic animal cancer and cystic fibrosis. Cystic fibrosis means gene related diseases. Okay. Cystic fibrosis and rheumatoid arthritis. Rheumatoid arthritis, that is the joint pain. arthritis. Okay. So this all the disease they are studied. This all the disease is caused by the gene. Okay. This all the thing was studied by using this transgenic animal. Okay. And they can prepare if this disease from this gene means they can know that this is a particular product of this gene, right? They can produce a medicine, right? So this is how this is very helpful in studying the diseases. Okay. Okay. Then we'll come to the bioproduct. Okay. So here we have taken from the transgenic animal, we have produced the alpha-1 antitrypsin. Alpha-1 antitrypsin. Okay. Alpha-1 antitrypsin we have produced. So this is useful in treatment of emphysema. Emphysema. What do you mean by emphysema, student? So in the emphysema, what will happen? So in the smoking person, it will be usually it will see the smoking person. Okay, smokers. So what will happen in the emphysema? The alveolar region is going to be decreases. Okay, so here the surface area in the emphysema, the surface area of alveolar is decreases. See here, this is the alveolar. So in the emphysema disease, here you can see the surface area was decreased. Okay. So due to this, what will happen? So there is decreases in carbon dioxide and O2 exchange. You know that, student, right? Where the oxygen and the carbon dioxide exchange will take place in the lungs, in the alveolar region, right? So because of that, there is a decrease in the carbon dioxide and the oxygen exchange. Okay. So now they have biologically, by using the transgenic animal, by inserting the antitrypsin gene in the animal, thereby they have get the antitrypsin protein. Okay. So now they have utilized it for the treatment of the emphysema. Okay. So this is one of the example. And second example is in the 1997. Okay. In the 1997, first transgenic cow was produced. That name is the Rosie. Okay. In this Rosie cow, first transgenic cow, okay, in this Rosie cow, it is going to produce the milk. This milk is very rich in alpha-lactalbumin. Alpha-lact, 1 minute. Alpha-lact, 1 minute. Okay, alpha-lactalbumin. Okay. So compared to the normal milk, this concentration was very, very less in the normal milk. Okay, it is very good for the newborn. Okay, it is very good for the newborn. It is going to produce 2.5 gram of the alpha-lactalbumin in the per liter of the milk. Okay, student, it is very, very, very useful, right, for the newborn. See here, in the milk, it is going to produce the alpha-lactalbumin. So that is the 2.5 gram per liter. Okay, so this is how helpful in the biological product. What they have did, they have inserted the gene in the cow. Okay, so that is required for the alpha-lactalbumin. So then in the milk, it is going to started to produce. Okay. Okay. So then let us study about so now we know how we will give the vaccine, right? So first we will give the vaccine. The vaccine was synthesized, prepared. First we will give that vaccine to the animals, right? First we will give that to the animals, right? We will not directly will give to the humans, right? It will give the reaction, or if something has happened, it is going to be leads to the death of that person, right? So first, whatever the things was prepared, first they will try that on the animals. So that is the mice. Okay. First they will try on the mice, and after the monkey, so then they are going to give to the human. Okay. So now they are trying to reduce the giving the trial using the monkey in the trial. So by preparing the transgenic mice, they are going to give that vaccine to the mice only. Okay. So thereby they wanted to reduce the giving vaccine to the giving the trial to the monkey. Okay, student. So that's why in the corona you have seen that first they have a clinical trial was happened. First they will go to the mice and then to the some other animals, then to the some small numbers of the people, and they are properly in the careful they have checked that people, they have checked the people whether they are having the any whether they will show any sometimes of the reaction or not. Then lastly they will give to the large number of the people. Then they will release that vaccine to the world, right? So this is how they will do the vaccine testing. Okay. Then coming to the chemical safety testing, for this what they will do, they will create the mice. They will create the mice that is very, very sensitive. Okay. For example, if the normal chemical is there means, for example, 1 minute. The normally this sensitive, okay. For example, normal mice is there, okay. Normal mice is there. For the normal mice, the chemical for the 1 gram of the chemical, it will show the reaction means, will produce the transgenic mice that will be sensitive to the 0.00.1 gram. Okay, so they will add the chemical. You show if this mice show any sensitive to that chemicals means they will not use that chemicals. Okay, for example, any medicine they have prepared, they will give to the sensitive mice. Okay, then they will give to the normal mice. Then they will use for the others. Okay, so this is how they will do the toxicity testing. Okay, they will by making the sensitive. Okay, okay. So this is about the ethical issues and the biopiracy. Let us talk in the next class. If you have any questions, you can ask me. Okay. Okay. So if you don't have any questions, means I will go for the NPS poll. You can give me your valuable feedback. Okay, student. Let us continue in the next in the tomorrow's class. Okay. You can give me your feedback. Yeah, then you can leave the class. We'll continue that in the next class. Okay, student. Thank you so much.